All right, it seems we are live. All right, everybody. So, this is our second rendition of Tissue Talks, and today, episode 2, we will consider Pogonia ophioglossoides a little bit more closely in depth. So, yesterday, we began our consideration of Pogonia ophioglossoides at the tail end of a broad overview of the Orchidaceae and things about a few other families. We're going to today focus instead. Let me just adjust this annoying chair. Much better. Today, we focus on Pogonia ophioglossoides, its life history, as well as its cultivation. So, as I said yesterday, Pogonia ophioglossoides is a small orchid. It's roughly 6 inches in height, and it has a relatively large 2 or 3 inch purple flower. It's self-compatible. It appears to be pollinated by insects, and it is a deceptive pollinator. Yesterday, we talked briefly about a complex of bog orchids, including Pogonia ophioglossoides, including several Platanthera, including the Calopogon tuberosus, Erethusa bulbosa, Calypso bulbosa, some of these things where, they're purple and there's like a splotch of yellow somewhere. And this fools certain insects because there's an aster of some sort in the wet meadows that these orchids inhabit that does reward its pollinators. The orchids don't give two fucks about their pollinators. They don't reward their pollinators. Some Platanthera do because they're pollinated by moths, and in order to coerce a moth, you do have to offer a moth a reward. Moths are relatively energy efficient pollinators, so they generally, along with hummingbirds, will drive selection for very rewarding flowers. Things that are pollinated primarily by moths and hummingbirds and a few other taxa have very, very sweet nectar, are generally edible, things like that, and they're usually relatively fragrant. Think like a nasturtium. Or there's some bog thing. It's got like a cup bowl shaped flower, and you can drink the nectar out of it. There are a lot of different things that are out there in these bogs, in these fens. As we established yesterday, Pogonia ophioglossoides is not all that particular about soil pH. It is only concerned with soil moisture. They have, among other monocots, a relatively efficient metabolic process. And this oftentimes means that, pineapple is another example of a monocot that's just really good at being metabolically efficient to some degree. And so Pogonia ophioglossoides doesn't necessarily rely on soil pH for things like nutrient availability. However, the reliance on different soil pH in a competition context may be important. I'm not entirely sure. The Pogonia doesn't necessarily need a specific habitat as long as it's really wet and really well illuminated. That's really all it needs. And so these things will rapidly and completely colonize any area of suitable habitat. They generally do it asexually as well. Okay. Now, we are going to, in a few, talk about the germination of Pogonia ophioglossoides, but we're not going to go there just yet. We do need to first consider a few things about the ability of this plant to colonize a particular habitat. And you would think that a habitat that's really, really wet and a habitat that's really, really sunny is going to be prime for most things. This is true. But something that happens frequently because you find some of these bog orchids in like wet areas that have like 10, 20-foot tree saplings, which indicates that there was this area that was disturbed maybe 50 years ago, maybe 100 years ago. I don't know. And then trees began to recolonize, but in the meantime, this flooded bog or fen, bogs and fens are both essentially high organic matter flooded wetland areas, but a bog has a low pH and a fen has a neutral or high pH. And so, there are some plants that are obligate wetland plants that you're only going to find in bogs. Think like your Sarracenia and other carnivorous plants, or fucking royal fern, or ostrich fern even. And you've got your what's that other one that only likes Cypripedium macaulay only likes very acidic areas that are well drained but moist constantly. Things in the Ericaceae, all of your like wintergreen and your fucking those cute little what are they? There's a lot of different stuff. Your mountain laurel and sheep laurel, they only like really acidic soils. These are soils that are associated not only with high organic matter, but a really rapid turnover of that organic matter into just essentially humus, because decomposition releases acids and that will just lower the pH. I got into an argument with somebody at one point who said, oh, well, you know what? Pine needles are useless because they don't lower the pH of anything. And I said, well, actually, they do lower the pH. Now, do they lower the pH more so than any other organic material? No. But inherent to being no longer living organic matter, those pine needles are breaking down, and that process is producing acids, and that is lowering that pH of that soil. So that high turnover of the organic material that you're seeing is characteristic of a bog. Okay. Now, there are plants that live in fens and fens only. Certain members of the Primulaceae, for example. I can't remember the exact one off the top of my head, but it's okay. Now, or am I thinking Lythraceae? You know what? It's not the end of the world. There are plenty of obligate fen plants. If you really want to know, you can like Google it. We're okay. Because one of the problems with obligate versus facultative wetland status is it's not necessarily indicating if the plant is obligate to either bog or fen, because you have plants like Pogonia ophioglossoides that don't give two shits if the pH is low or if the pH is high. As long as it's very wet and the sun is like beating down on it, okay? As long as those two criteria are met, those plants are going to do 100% fine, okay? And we need to understand that in some of these areas, right, you have plants that compete aggressively for space. Some of the sensitive fern comes to mind. Anoclea sensibilis is this interesting looking, very cool, but quite aggressive as long as it has enough water. And I need to find some. Where did I put the water? Um, so I could indulge my really bad pathological demand avoidance right now and not seek the water that I'm craving, or I could be a responsible adult person and look for my. I'll be right back. Water. Where the fuck? I'm doing the fridge. I'll take the water. You know why? Itchiness. I have managed to locate my water. I have very dry mouth. So I'm going to take a sip of my water and then we're going to talk about water. Yeah, perfect. Now, something to consider about the interaction of water and sunlight with the addition of organic material that has now broken down to the point in which, um, you know, it's now its nutrients are now accessible, bioavailable to plants. So one of the things that gardeners talk about, and it is very much true, it is backed in science 100%. Oh man, sorry, my eyes are itchy. Anyway, now a lot of gardeners will talk about this thing where if you have fresh manure, fresh compost, and you put it on plants, it will burn, quote unquote, burn the roots, and that may entirely kill the plant. Now, what this is a reference to is the fact that when compost manure type material is that fresh, the nitrogen in it is organic, but it is in the form of ammonia and not a nitrate of some sort. Now, because of that, ammonia is highly toxic to plant cells, especially the root cells, not as much to like the leaf material, but ammonia in high enough concentrations will still kill plant material. But the nitrogen source can be too toxic for the very delicate root meristem and material like that, you know, the tender young material, the roots. And then the plant is shock wilting and having some issues where it now has to recover from that. And so that happens because the nitrogen is just too aggressively bioavailable at that point to very young material, and it destroys it. Now, if you age compost manure, things like that, for, 6 months, a year, something like that, the bacteria and it convert the nitrogen further into a form that's more acceptable to the plant. Okay. And because of that, aging compost and stuff like that, that wisdom from gardening and things like that is totally correct. It is something that, the science is clear on. So, anyway, Pogonia ophioglossoides is a little bit weird because it, to some people's assessment, is primarily an asexual reproducer, even though it still bothers to produce seeds in the fall. So this has me thinking about a couple of things because I have a little bit of a hunch on a couple Pogonia related thingamajiggers, and we're gonna get into it. But how do I put this? If you were to go to a fen or to a bog and you were to, you know, find an area where these orchids were growing, okay? Now, what I will put a, disclaimer, I don't condone the poaching of wild orchids. However, if it is, you know, the collection of just the propagule material so that you may have it in your collection. So in many species, that would be the seeds. In some species that don't germinate well, it may be a division or something like that. That I would generally find acceptable as long as the material is not being sold, collected for the sole purpose of selling, things like that. You have to understand that one of the ways that we learn about orchids and help their conservation and appreciate them better is to cultivate them. And there is a point where you do have to obtain wild propagules of any species you are wishing to work with in cultivation. Orchids are no exception to that. So, in a nutshell, and the other disclaimer, there I do not have as many qualms about what people do to the orchids or any plant on their private property as on like wild grounds, whatever. So, if you have a backyard, why am I saying this? If you have a backyard full of, Cypripedium, and you want to mow them down, God, why am I like this? No, but seriously, that's your choice. It's your private property, whatever. So that's the other disclaimer here. So, like, I have had people give me orchids collected, like dug up from private property before. It's not ideal, but at the same time, you have two choices. You either like get that landowner, the homeowner, whatever, on board with like making sure that plant is okay, or you try to get it the fuck out of there to somebody who may be more competent. And in some some situations, like my oh god, I'm not even going to talk about my neighbor's yard because they're going to get mad at me, and I don't want them to get mad at me for that. So yeah, we're not even going to go there because it's not relevant. It doesn't really matter. But in any case, private property is a different matter than, hey, we're out at the wildlife management area. Let's rip up all these orchid things. No, don't do that, people. I will generally make sure that if I'm going to collect seeds from anything, there's not like only one seed pod there. That would be really, really bad. Just let it be. Unless it's like a very endangered thing that I'm very good at propagating. And I'm very good at propagating a great number of different things, but I don't think, other than different Lycopodiaceae, we'll talk about Lycopodiaceae again in a different episode, okay? I do have a Rearing Rambles episode, an earlier Rearing Rambles episode on some of the Lycopodiaceae. So if you're very curious about those Lycopodiaceae, you can go to that other one. So anywho, what ends up happening is people are, really nervous because they're selling their property and they have like this incredibly rare orchid on their property. Now, one of the things that you have to understand about the vast majority of native orchids is they don't respond to being toyed around with, move, whatever, when they're in active growth all that well, okay? So if you can do your digging the thing up when it's dormant, that's going to be a lot better for the plant, okay? Now, if that's not possible, I wish you the best of luck. It's kind of a coin flip at that point. If anybody has, any type of advice, any tips for us on like, hey, I have this method of like transplanting cypripedium while actively growing that doesn't kill them, please let us know. Like, that would be very, very useful information. Even if it's anecdotal, at this point, we're kind of just, we need whatever, we need anything. But anyway, you know, I say all this with love. Everything is going to be just fine. Now, back to Pogonia ophioglossoides. This is an incredibly widespread, aggressively growing orchid. Going in and, lopping a small fragment of rhizome off at the edge of like a colony is not going to do jack shit to harm or disrupt that population. As long as, you are not like using a filthy knife that's transmitting a disease into the, as long as something like that doesn't happen, you're probably almost certainly fine taking a small segment off of the edge of, the edges of these colonies have to be able to withstand, like, hey, some deer just, fucking rammed its hoof right down into the edge of us, and now Jimmy Joe Bob over there was crushed. No, whatever. They have to be able to tolerate that to some degree, or they will be fucking wiped out. Having accidentally trampled. I've trampled garden plants. My dog has trampled garden plants, and they've been fine. It just happens. So like, that's not a horrible, like, morally abhorrent thing to do. Now, other than that, that's a really good way to start, because what's going to happen is the plant basically has this rhizome, and it kind of pretzely, kind of just corkscrewy, kind of wriggles through the substrate, and quite a bit often, it sends out this clump of root material and a shoot. It's a typical monocot style, kind of like a couple of like onion rooty, like little thingies that go down, and there's like a single stalk, and there's a single leaf, and these things have got to be like horrendous or like magnificently metabolically efficient, because it's like a single little leaf. But again, they're getting a shit ton of water from below, a shit ton of sunlight from above. Other than like several soil minerals that they're going to need, and maybe some organic nitrogen, they're ready to roll. The other thing about the Pogonias is that their small size means that their impact on the resources, especially within the soil, the substrate the plants are growing in, is confined to, the very top layer. A lot of things that are maybe even just a slight bit larger in stature than Pogonia ophioglossoides have their roots far below where Pogonia ophioglossoides can venture at all. And it is precisely because of that Pogonia ophioglossoides kind of just is more or less a master of its own domain. Now, it's a really pretty little plant, and other than the fact that it will overtake a pot very quickly, under the right conditions, it is really a nice little I would love to have it as a ground cover, for example. Wouldn't that be awesome to have like some flood yard, and it's just a bog full of Pogonia. I mean, like, that is a thing. Like, it's a cool plant to look at, but like, I don't necessarily need to see 84 quadrillion iterations of it. I'm sorry. I mean, it's really pretty and it's really cute, and my limit is maybe a couple million, but in any case, it is a very lovely little plant. So, how does it work? Pogonia ophioglossoides is a plant that generally emerges from the substrate a little bit later than some other plants in the region. Especially for a bog plant, you generally don't see them popping up until like the first week of June, sometime around there in our area, in New England. And, I'm going to kind of go out looking for them later, maybe tomorrow or whatever. But anyway, I know a couple populations of them, and I adore them very much. So I got to go see the Pogonias. But other than that, we will now get into tissue culture, because we have established that you can very easily just kind of clone a fragment of this Pogonia colony, and it kind of just reiterates itself as it wishes. Okay, it's not it's not as obnoxiously aggressive as something like the sensitive fern Ranunculus sensibilis. I know people who are like constantly trimming it back quite a bit because it is incredibly vigorous. But Pogonia ophioglossoides doesn't necessarily have the most obnoxious way of coating the surfaces of things, but it certainly is a very rapid developing plant. Now, you may be wondering then why on earth I'd even be concerning myself with getting the seeds to germinate in tissue culture. And I am very glad that there is somebody who may ask me that question. I have to say, that is something. One of the ways I need to entertain myself every now and again is by being so absolutely asinine, so just incredibly extra about something really trivial, such as trying to germinate Pogonia ophioglossoides seeds in tissue culture. But we will talk. Now, what I will say, I will justify very quickly why you might want to germinate the seeds of something that you don't really need to germinate the seeds of in order to propagate. And that is because it's not always just about propagating, period. It's also about understanding the natural history of the organism and the value of having a means of sexually propagating it is, let's be honest, one of the easiest ways to ensure you can rely on some genetic recombination and variability, because you never want to rely on a monoculture, especially a genetic one. Gotta say it, okay? But in any case, if indeed it is important to have some control over our genetic repertoire for as many species as we can, especially if conservation is the goal. And what I'll say, there are places in the world that have cataloged literally as many seeds of as many species of as many plants that we know of as they can. Probably also spores and things that have non seed plants, because not every plant's a seed plant. Anyway, um, you have to understand that our understanding of biology is such at this point that we don't understand enough about species interactions to rule out the importance of any particular species. And so there is some value in having a backup for what if we lose a species. You have to hope that that never happens. Now, one of the things you also have to understand is that a lot of species that there are plenty, don't necessarily rely on how common a species is right now as an indicator of how common it will be in 50 years or something of that sort. You have to understand that there are plenty of species that were once common that are now rare or extinct. Okay? And also, common species are rare. And what I mean by that is there are some species that are keystone species in the sense that relative to their biomass, they play a quite disproportionately large role in their environment. So apex predators are oftentimes regarded as keystone species. Now, there are also species that are foundational species, and these are oftentimes the species that are highly abundant, sometimes obnoxiously abundant. Okay? Obnoxiously abundant things tend to be very good food sources. What you have to remember is, I'm trying to take a sip of water. What you've got to remember is that when an organism is developing, it only has so much energy to develop to one thing or another. Okay? Organisms that put a lot of energy into tasting like absolute garbage, or eating something toxic enough to kill their predator without killing them, or I don't know, having a really tough outer shell, something that takes a lot of energy away from developing eggs and sperm. Okay? And so organisms make up for that either by being more slow to mature, giving themselves more time to reach a suitable size so as to have a reproductive event that is quite large, producing multiple offspring in one round of reproductive activity, or they have multiple very small bursts of reproductive activity. Now, you'll see in some insect groups, I don't want to, well, yeah, we can quickly talk about insects. In mantises, for example, you have some mantis species where the female lays a great many very small egg masses that have small numbers of eggs, or you have species where the female lays just a few egg masses, but they're very large and hatch several hundred nymphs at a time. And both strategies are just different life history strategies. Now, if you have a romanticist, personally, I preferred, things that didn't hatch out 40 bajillion fucking little goblins at once. I preferred things like Creobarata were fine because they seldom hatched more than like 60 nymphs at a time. And that was kind of my upper limit on like hatch numbers. Because anything other than bigger than that was just a real pain in the fucking ass to try and deal with at once, especially, sometimes if you're not careful in like how you time your fruit fly cultures, it can be a risk doing like large batches of fruit fly cultures all at once versus having, a few cultures made every week. And you can kind of like do both strategies because sometimes some batches of fruit fly medium get really fucked up or something is weird. Fruit flies aren't perfect organisms, and in any case, things that had a more modest hatch rate just fared better because, I would be able to ensure more carefully that each nymph was given enough nutrition in its earliest days. It's a lot easier to feed fruit flies to 100 nymphs if it's 20 nymphs a week over 5 weeks than if it's 100 nymphs all at one time. In case I hope that makes sense to everybody. And so there are plants that are kind of the same way, and there are these just different mixes and matches of strategies. But what I'm getting at here is that something like Pogonia ophioglossoides, that's super, super common, is found all over the goddamn place as long as it's wet and sunny enough, because it puts all of its energy into just making copies of itself, like a copy machine. Just boop, 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 boop. But those plants are relatively like soft and squishy and small and yummy, yummy. They're not well defended against, raunchy herbivore creature things, and that's fine because there's no way one deer or one if I decide I want to taste of Pogonia, no, I'm just kidding. Or a dog, because dogs sometimes eat orchids. And nothing is going to eat the entire colony of Pogonia ophioglossoides. And by the time something else comes to eat any more Pogonia ophioglossoides, they've all grown back. It's that sort of thing. But it still bothers to produce flowers. And it still bothers to produce seeds that have embryos inside of it. Although, several people, I don't want to dox people out. I mean, I just don't want to name names unnecessarily. But several people in my circle and I have really no success to speak of with germinating these seeds in tissue culture. Now, I was looking on Google Scholar at a couple of things yesterday, and we're going to talk about that a little bit in just a moment. But I have a hypothesis about this. I suspect that those patches of habitat that are ideal for Pogonia ophioglossoides don't last forever. Surely they don't. No habitat type lasts forever in one area. You people understand that there's something called succession. So, that area of like humongous trees, right, where everything is like 200 feet off the ground, and it's just like the forest floor is covered in like rotting logs and fucking there's owls and there's like a foot of leaf litter on, that's really old growth forest. Well, at one point that was bare ground. Many hundreds, even thousands of years ago, yes. Maybe right after the last glaciation event. But what I'm getting at is, to a degree, a lot of habitat types are just different steps in a process where an area that had no soil at one point gets soil on it. Okay, and that happens by a rock material eroding, facilitated by water, as well as lichens and things of that nature. Okay. And that process makes soil, and then other organisms start to enter the area over time. And so then you start to get some, like, you might get like a grassland habitat or something of that nature, like a meadow. And some areas that are more prone to like a higher water table or something of that sort might escalate into like a bog or a fen, And so this is all just different steps along just this gradient of things that happen. And it's just referred to as succession. And I'm aware I'm oversimplifying it. I don't have the most extensive knowledge of succession. I have a very basic understanding of how succession works. Now, because the majority of things that I do in my research doesn't necessarily rely on the successional stages of an area, I'm not as concerned about ecological succession as somebody who may be concerned about ecological succession for something such as habitat conservation, restoration, ecology, whatever. That's just not my subdivision. I do a lot of things like nutritional ecology as well as monocrop physiology right now. I'm not the person to go to if you're curious about something regarding, say, what's it called? The successional ecology. I basically understand that there's primary and secondary succession, and basically, because succession is how an ecosystem progresses after a disturbance. And along with that, there's the intermediate disturbance hypothesis, which is based on this idea that there are habitats that are very rich because they receive sufficient disturbance to prevent the only organisms from that are in the area from being really, really competitive. But it's not disturbed so harshly or so frequently that only the most well-defended organisms can survive there. This is part of why, they say to not necessarily wash your hands with like soap and hand sanitizer and everything every time you wash your hands, or every 30 minutes. You're not supposed to do that, for example. And part of the reason for that is because your skin does have a distinct gut microbiome, and your skin actually does require bacteria. Your skin should not be like fucking sterile, completely devoid of non-human cellular life. Okay? What you have to understand is that there are various microbes on our skin that help us, combat the damage from the UV rays in the sun. They help us. They absorb some of the metabolic byproducts of the skin cells. They just do things that are beneficial to the health of the skin surface. Okay, you don't want to constantly strip your skin. Now, I'm not saying, put your bare hands and disgusting whatever. And I'm also not saying don't wash your hands well if you get them contaminated with, for example, feces. I'm not saying that. What I'm saying is, if you're, if you haven't done any like major, like anything that gets your hands soiled and dirty, you probably don't need to use a shit ton of soap and water before you pick up a carrot and pop it in your mouth. Come on, just be a little bit more reasonable, because you don't need to continually remove shit off the surface of your skin. Anyway, intermediate disturbance hypothesis. When an area has no disturbance, it's just the rapid colonizers that don't give two shits about, if something eats them or like dies on top of them. They do their thing and grow. If the area is just continually getting bombarded somehow, then the only things that can live there are things that don't give a fuck if they're like, set on fire and then like, flooded and like under 8 feet of snow for 3 months. And those things can't withstand all of that without being very slow to develop, because they need to invest so much energy into not dying, okay? So, Pogonia ophioglossoides is a species that does really well, but its habitat isn't permanent. Now, recall yesterday that there was a consideration of the size of the orchid seed and how it's incredibly tiny. It's wind dispersed, and it goes ahead and it can, it needs to land somewhere really well. So anyway, remember that orchids do this. Now, the TLDR of what I gathered yesterday or the day before, I can't remember. No, sorry. It was the day before yesterday, not yesterday, the day before. My bad. What I gathered from some of my research on Google Scholar was that the embryos of Pogonia ophioglossoides are just in some dormant state that is prolonged. And some of the authors of some of these papers suspect that there's something that is just allowing the dormancy to be delayed. And that is kind of a characteristic of something that is adapted to entering the seed bank. Now, the seed bank is basically this, just, you have soil, and if it's soil that's been outside, it's going to have seeds from whatever plants were nearby. Now, often, not always, but there will be a great number of those plants whose seeds can stay hidden away, dormant for many, many years in that what's called a seed bank. The soil, seeds that need to like hit the light to germinate are things where like, okay, the seed has been buried, sassafras and a number of other things, oenithera, some other things. The seeds need light to germinate. And so what happens is when the seed bank is in a disturbance of some sort, if you know, take a bunch of dirt from like the bottom of your compost heap or something, and you put it in like a tray and water it, in a couple of weeks you're going to have little seedlings come up, even if you have it inside. And that's because there were seeds in there. And as soon as enough light hit them, they were at the surface of that substrate, they germinated. You didn't see them because over the years they have taken on the color of the soil, but they were there. We've done it before. We've literally scraped like the top layer of like dirt off of this pile of like sandy dirt shit. And then we scooped some of that soil into some trays and gave it a good watering, put it in a greenhouse. And yeah, we had just all sorts of grasses and other little things, dicots of whatever flavor, just kind of coming up. It was so interesting. This is what a seed bank is, folks. Now, I just suspect Pogonia ophioglossoides is one of those seed bank species, even though it's an orchid. So, man, I'm feeling quite tired, but that's okay. What we're going to do is I'm going to take just a very short break. And then what we're going to do is we're going to talk a little bit about the in vitro propagation of Pogonia ophioglossoides. Or, well, let me think. Yeah, I think we'll just take a quick break. All right, let's see. Oh, much better. How about all right? So just to check a couple quick things. Not the end of the world. Ugh. All right. Ugh. All right, people. So I am quite tired. We're gonna get through this together, okay? All right. You know what? I think we're gonna call it here because I'm exhausted. But stay tuned. Part 3. Oh, somebody joined, but unfortunately, I am ending the stream now because I am just a little too tired to go on. But I will finish it in a day or two, and we'll have a fun time doing it. Now, other things we will do later in the season are various ferns. That'll be fun. And yeah, so just kind of look forward to a few things. One of the things that I might do is just have the first Pogonia episode kind of lay down a couple fun. Well, yeah, we're gonna lay down a couple foundational things about orchid propagation and stuff like that. That might be important for future episodes. We kind of did that in the Akali episode. I mean, I have other stuff on my channel about the Akali propagation. I think there's, if you were like to be enough creepy stocky on my videos, you could find all you wanted to know. Like you would see me like pouring plates and replating proto cow worms and everything. And I might even like try and make a tutorial that blind people can. No, just kidding. I I don't know. I wonder if a blind person could do tissue culture. I feel like that might be one of the only things that you really do absolutely need your like eyes to, especially since it's like a really delicate thing where like if you're doing a lot of tactile shit to your things, you're probably gonna get contamination. Um yeah. Anyway. Yeah, I think I'm going to just end the stream here.